Okay, everybody, we're going to get started, and anyone else can just trickle in later. So we're really excited to have Dr. Doug Sponsler here today. Doug is a postdoc in the Center for Pollinator Research at Penn State University, but also a research associate here in the botany department. Doug got his PhD in entomology from the Ohio State University and his bachelor's in biology at Cedarville University, which is also in Ohio. His research is focused on bees, as you can see on this next slide. He studies what they eat, how they're affected by urbanization and farming, and even how to interpret their dances. Outside of his studies, he is an administrator for iNaturalist, which looks like it's a site where scientists and members of the public can kind of share information about what they find in the outside world. And maybe you can tell us a bit more about that later. That sounded really interesting.
I will, in fact, in this talk.
Oh, wow, that's cool.
Though truth be told, I'm not an administrator in the technical sense. I administer particular projects, as any of you could as well, if you create one.
Oh,
So,
well, I'm
yes,
supposed to learn more about that, so it's just from my CV reading.
Yeah.
Anyway, with that, we are delighted to have him here today to tell us more about what he does.
Well, hi, folks. Thanks for coming. It's a pleasure to get to present to you today. It's also a pleasure, I must say, to get to present a 35-minute presentation. Yesterday, I presented a 10-minute presentation, and I realized that it's actually the first time I've had to do that in years, like since being a grad student, and it is so hard to give a 10-minute talk. So it's good to be back in the more expansive world that will accommodate my rambling style much more comfortably. The premise for my talk today is the ecological centrality of flowers as food. So one of the most salient macroevolutionary stories that we can reconstruct is the one pictured in this plot here from La Bandera, 2014, which shows the transition between from a world in which plant-insect interactions were dominated by gymnosperms to one in which plant-insect interactions were dominated by angiosperms. So insects have gone from interacting with gymnosperms to angiosperms, and the shift has been dramatic to the point where the vast majority of plant-insect interactions today are interactions between insects and flowering plants. One way to interpret this, as suggested by Dawn Frame in her 2003 paper, is that what angiosperms have effectively done is harness the maladaptive phenomenon of herbivory into an adaptive phenomenon of florivory, or it is still herbivory. So when we see this bee, or in fact this beetle, visiting this flower, this is an act of herbivory, right? But it's a specialized act of herbivory by a specialized herbivore upon specialized parts of the plant. So it is a harnessed herbivory that is now adaptive for the plant insofar as it facilitates pollen transmission. The effect of this has been dramatic. In a recent paper, Glenn Wardhow estimates that there are about 352,000 species of arthropod that visit flowering plants. If you add this to the roughly 369,000 species of angiosperms, you get about a third of all extant described eukaryota on Earth involve this floral insect trophic interface. So that's all well and good theoretically, but can we gain any empirical traction on this? I think that if we zoom in on this floral trophic interface, we can capture a lot of it with a model like this. So what we have here is an axis of resource volumes. You can imagine this as being the total amount of like the commodity resource availability from flowers in a given place. And this, of course, is going to vary through time with the phenological progression of the plants. Then for any slice of time along this curve, you have a taxonomic matrix consisting of the floral taxa in bloom at any given time and their relative abundances, which will also vary in time. And then both resource volume and resource composition are going to vary in space. So we have these four quasi-dimensions, if you will, of space, time, volume, and composition. I'm going to suggest a term here for this, floral resource topography. In coining this term, what I want to do is evoke the sense of a multidimensional surface of sorts that's composed of the states that these dimensions of floral resource availability take. And my proposal is that if you understand the shape, if you will, of the floral resource topography of a place, then you know a lot about the architecture of biodiversity that underlies that place. I have to admit, I became attached to the term floral resource topography before it occurred to me that there appears to be only one way to pronounce the acronym, which is not what I want to be known for. But, okay, so I think it's a helpful model, but is it empirically tractable? This is not a new question, in fact. This question was posed quite explicitly by Frankel et al. In this 2005 paper. A paper that, for whatever reason, has gone almost entirely uncited in the years since, even by its own authors. But in it, they pose the question of whether quantitative floral phenology at a landscape scale is possible, recognizing that quantitative floral phenology at a landscape scale is foundational to our understanding of the biodiversity of flower-visiting insects, especially pollinators. Now, one way to approach this, which is kind of like the way the original authors did, but I think is kind of epitomized in the work of my friend Aaron here, who did this during his postdoc at Cornell, is to go out and actually do old-fashioned floral surveying and phenological scoring over vast areas, running huge transects, doing quadrat sampling. This requires extraordinary botanical expertise, which Aaron has, and I do not. But it also, you still run into limitations of how much area you can actually cover with this approach. Moreover, if your research system looks like this, you encounter other difficulties, like, for example, running a transect through downtown Philadelphia. You would most likely be hit by a car in the process of your sampling, right? There's private property, there's built structures. In urban systems, and perhaps some other landscapes you could imagine, traditional floral sampling techniques are rendered virtually impracticable. So, is there an alternative approach? My contention is that there is. This is where I will introduce what has, I think, regrettably become something of an insecta non grata in the ecological world recently. But the European honeybee, granted an introduced species, but one that we owe quite a lot to, is arguably the most remarkable foraging organism in the world. It forages at a scale of routinely several kilometers from its colony, resulting in a total foraging area of routinely 100 square kilometers. So it forages at a landscape scale, and it does so in great numbers, and conveniently returns the results of its foraging to a box that you can set up and then sample. So I harness the sampling power of the honeybee with the help of my friend Don, who's the man behind the bees here. He runs the Philadelphia Bee Company. And we used the apicultural infrastructure he already had in place in the city to sample Philadelphia's floral resources through time and across space, trying to capture the internal heterogeneity of the city and sampling it over 2 years in 2017 and 2018 from spring through fall. And what we did to capture all the dimensions of the floral resource topography of Philadelphia, which, by the way, is also my hometown. While I went to school in Ohio, I grew up here, and so it's been a homecoming to come back and do research in the city. To capture the volume through time dimensions of floral resource topography, we used continuous colony weight monitoring with data logging hive scales. So you set up these scales under your hives, they record hourly weights. And then with a pipeline that I built in R, we can clean up these weights and we can model them using generalized additive models to create smooth weight curves. And then ultimately what I do is take the first order difference of those smooth curves, so you get a curve that oscillates above and below zero, reflecting weight gain or weight loss. And I'll show you those curves later. Then to capture the taxonomic dimensions of the floral resource topography, we use pollen DNA metabarcoding. Specifically, we use a multi-locus approach. So with next generation sequencing, there's always a fuzzy relationship between sequencing read counts and the actual quantitative composition of your sample. But you can mitigate quantitative bias by using multiple markers and making your quantitative inferences based on the median read counts across multiple markers. So we used 3 markers in this case. This is from an earlier paper we published, but for this study we used ITS1, ITS2, and TRNL, for those of you familiar with the world of barcoding markers. And then finally, we used waggle dance mapping. This is one of the other reasons why honeybees are the most remarkable foraging organism on the planet. They're the only organism besides humans, I suppose, that can tell you, and in fact do tell you, where they went to get their food. And you can interpret that and use that to map spatial patterns of floral resources. And to do this, we used the probabilistic method developed by Roger Shirk in his 2013 paper. This is that method adapted in an earlier paper of ours, and I'll show you some data later from Philadelphia. But it plots dances as probability clouds rather than discrete points, because that would exaggerate the precision of the dance. And to improve the efficiency of this, I built a device that attaches a Raspberry Pi computer to a camera. So it's like a $25 computer attached to a $25 camera. And it's Wi-Fi connected, so you can run multiple cameras across multiple sites in a synchronized way, and allows you to collect synchronized data without having to physically be there turning on a camcorder. So this was set up in the Outside In exhibit. Very grateful to the exhibit staff for letting me do that. The hope is to do it again next year and get a fuller season of data. The data that I'm going to present from this are very preliminary, but it was a proof of methodology mainly, but I do have some cool pictures to show you. All right, so and this is what our site layout looks like. While I'm not going to present a landscape analysis here, there is an extremely high-resolution land cover data set available that covers our area, recently released by the Chesapeake Bay Conservancy, that gives a 1-meter resolution in a classified landscape layer that's basically like the National Land Cover Database layer, except at 900 times better resolution. So, which is required for urban areas, because at a 30 by 30 meter resolution, like typical data sets are, something like a street tree is going to be completely invisible, even though it's a significant part of urban green space. All right, so getting into the results of this study, I'm going to be showing results from across the 2 years, 2017 and 2018 of this study. The gist of what we saw in terms of a global model of the pattern of resource availability across these 2 years was actually rather consistent. We see two strong periods of weight gain during the spring, one in May, one in June. Again, this is first order difference data, so when the curve is above 0, it's weight gain. When it's below 0, it's weight loss. 2017, we didn't sample as early or as late, so it's a bit truncated in either direction. Then as we go into July, weight starts to flatten off. In August, every single colony in our study system lost weight. In September, we see a brief but strong pulse of resource availability where hives are gaining weight again. And then that's effectively the end of the foraging season. And from mid-September to frost, colonies are losing weight. This is what it looks like when we plot curves for individual sites. So each panel here is a site. This is the integrated data. This is the first order difference data. This is the global model I showed you before, integrated and differenced. And these are the site-specific curves. And you can see that there are site differences, but there's a strong evidence for a global model. And in fact, I fit this with a hierarchical generalized additive model, and via model selection, the model selection supported the use of a model including both a global consensus curve and a site-specific model component. So that suggests, albeit circumstantially, that there is a significant site effect within urban areas, which was virtually a premise of my study, but it's good to see it borne out in the data. And as I continue to analyze the data, I plan to do more spatially explicit analysis. In terms of resource composition, given the time, I'm not going to get into the actual taxon by taxon commentary, though that's what I'm always tempted to do, because each taxon is interesting in its own way. But I want to show you some more aggregate data. This is genus richness. I should say, too, the barcoding that I'm doing is to genus level. I don't feel confident going to species level with the data that we have available right now. In the literature, you will find groups that claim species level barcoding. I think that's an under-conservative approach, so I'm going to genus level only. But interestingly, the diversity roughly mirrors the patterns we saw in the weight, right? We have peak diversity in May and June, declining in July, reaching a minimum in August and October are similarly low, and then there's a little bit of a rise, granted not a statistically significant one, in September, which suggests that there's a relationship, perhaps not a causal one, but a relationship between diversity and availability, that when a lot of resources are coming in, it's a lot of different resources. This is one of the findings I find most interesting. So this is a principal coordinates ordination. It's related to principal components analysis, if you're familiar with that technique. It's basically a dimensionality reduction technique that allows you to plot high dimensional data in 2 dimensions to try to explain all the variance you can. In this case, the sample period is colored, and then the year are shown by the type of symbol. One thing that jumps out immediately is that the years were actually, I was shocked at how consistent across the 2 years our data were. The pollen, the community composition falls right on top of each other across the years within sampling period. But what I find most interesting here is the discontinuities we see here and here. We have kind of a continuous flow of phenology, but then there are these disjunctures that I think are ecologically significant. And if I could speculate on an ecological explanation for this discontinuity between May and June probably reflects canopy closure. That's when tree canopies close. It's the end of tree flowering for most species, and also the end of the spring ephemeral flowers that grow under trees. And with it, we have a shift in flowering phenology toward open habitat taxa, and a corresponding shift in foraging animals to those taxa that are in bloom after canopy closure. The second big shift, though, this is driven mainly by the fact that trifolium is probably the single most important resource in our area from the perspective of foraging honeybees, anyway. And trifolium bloom starts it blooms practically all year. It starts a little bit in May, and it will go till frost, but its peak is in June, July, declining into August, and then by September, there's very little trifolium left. And so part of this is due to the decline in trifolium in our samples. So this is white clover and hybrid clover and red clover are all trifolium species we have here, as well as a few others. But then when we pick back up again in September and in September and October, the flora is suddenly dominated by these weedy woody vines, exotic woody vines, clematis and hedera. Clematis ternifolia in this case, the sweet autumn clematis, and probably some combination of hedera helix and hedera hibernica, which are both exotic ivies that are. What I think we see here is actually a characteristically urban flora. Interestingly, if you go back and you read the classical literature about bee flowers, because I'm not the first person to ask these questions, people have been interested in what flowers bees have been foraging on for a long time. If you look back at that literature, you will find no mention of either clematis or hedera. Recently, there's been some appreciation for hedera in England. But I think what this reflects, there are 2 possible explanations. One is that traditionally, urban landscapes are systematically neglected in ecological thinking. And I suspect that people were just not looking for what bees are foraging on in cities, where the particular disturbance regime really favors these colonizing woody vines. It's also possible, though, I think it's especially true of clematis, which is a more recently been elevated to invasive status, that the abundance we see reflects more recent invasion success on the part of these vines. And I should say the classical literature I'm referring to is mostly from the 1920s or earlier. So both of these species were present in North America at that time, but perhaps not with the level of dominance that they are currently. Then if we look at trait analysis of resource composition, the caveat here is that I got these traits from the USDA plants database, and I only assigned a trait to a given genus if all of its constituent species for our region were uniform for that particular trait. So whenever you see a big block of undetermined here, that means we had non-uniformity within the species of a given genus. But nevertheless, some salient patterns emerge. With respect to growth form, we see an early dominance of trees and shrubs giving way to herbaceous plants, as I said, accompanying that shift after canopy closure. We see it in both years. And then there's in August, again, we see a resurgence of trees and shrubs. And then this here is that dominance of woody vines that I mentioned earlier that is forms the vast majority of the foraging environment in the fall. Note the near total absence of annuals and biennials from our samples if we assume that the undetermined taxa reflect the distribution we see in the determined ones. This supports the notion that a lot of the kind of aesthetic annual gardening that people do, oftentimes with the best of intentions, is essentially ecological decoration. It's not participating trophically. Now, this is only honeybee foraging that we're actually using here. It's possible that annuals are being visited by other insects. It's also possible that they are providing only nectar and therefore not showing up in our pollen data. But I still think this pattern is striking. It's really perennial plants that are dominating the overall foraging landscape. And then the pattern of native versus introduced taxa. We see a majority of native taxa only in the spring, and that's because that's when the native canopy trees are blooming. Once they are done, it is mostly the type of plants that colonize open disturbed landscapes, which tend to be exotic plants like the clovers that we saw, like many of the woody vines that we saw, and like many of the ornamentals that start to become important later in the year. I want to show you some pictures. These are all pictures that I've taken in Philadelphia riding my bike to work, so these are relevant to the study system. And this is going to go through the season in chronologically, so you can get a feel for the plants that we're talking about here. One thing I want you to appreciate is the importance of wind-pollinated plants as pollen sources for insects. There's a traditional assumption that if a plant is wind-pollinated, it is not of trophic importance to insects, because why would it be? It's not in the plant's interest to attract insects. However, it does not mean it's not in the insect's interest to eat the plant. And so these plants are probably not, for the most part, producing nectar. Though some, in both these cases, both maple and willow are kind of in between in their pollination biology, and they do rely somewhat on insect pollination and produce some nectar. But even plants like ash here or London plane tree, which are, as far as we know, strictly wind pollinated, are still major resources that we see early in the season for pollen, not for nectar. Alongside the wind pollinated plants, though, we also see the rosaceous trees, like I believe this is hawthorn. It's possible it's apple, but it's a rosaceous tree there. Now moving into late spring and into the summer, some plants that really dominate our samples include, most importantly, clover. This is Alsike clover here, Trifolium hybridum, though also Trifolium repens and Trifolium pratense are important sources in this area. Over here, we have Parthenocissus, Virginia creeper, which I think is a resource. It's a native woody vine that I think is really underappreciated. It gets very little mention in the literature as being a resource for bees or for other insects, but it is covered with foraging insects. Note its rather drab floral display, though I think this is one of the reasons why it gets overlooked. From our perspective, there's not much to attract the eye there, but this is evidently a very attractive resource for insects. And then over here, can anybody, you would all be able to identify this plant if you saw the trunk. Can anyone identify the flowers, though? This is it's Aurelia alata is the Latin name. The devil's walking stick is what it's commonly called. It's that spiky tree that when you're walking in the Wissahickon, which is where it primarily grows, when you slip and you reach out to steady yourself, what you grab is invariably one of these thorny trunks of Aurelia alata. It's almost identical to a native Aurelia spinosa. I don't know how to tell the difference, but I am told that the native Aurelia spinosa has been virtually extirpated by Aurelia alata, so I take people's word for that.
Did they use the plantain that's also next to the clover?
Yes, they do. Plantago is not as important a resource as the clover, not even close, but it does show up. And plantain is, of course, very common in in disturbed habitats. Some other important plants include verbascum here, common mullein, or perhaps giant mullein, one of the two. Verbascum is the genus. This is yellow sweet clover, Melolotus officinalis, which I believe yellow and white sweet clover have recently been collapsed into the same species, right? We don't recognize two species there anymore. So this is sweet clover. And then as we get later into the summer, one thing I thought was very interesting is the emergence of plants that are definitely ornamental. So I said earlier that the minor role of annuals could be interpreted as support for the common notion that the plants people plant because they're pretty are not ecologically functional. Well, I think the exception to that rule, and it's a very significant exception, is the woody plants that people plant for ornamental purposes can be extremely important as foraging resources. So this is Lagerstromia, crepe myrtle. This is Styphnolobium, Japanese pagoda tree, both of these exotic. This is hydrangea. I don't know what species, but we have both native and exotic hydrangeas. These two occur exclusively, virtually exclusively as planted ornamentals. My understanding is that Styphnolobium, Japanese pagoda tree, has naturalized to some extent and can be found escaped from cultivation in parts of the city. And then hydrangea, there are some wild hydrangea, but they're predominantly cultivated garden plants. So it is encouraging to see that some of our ornamental landscaping is worth something ecologically. And then when we get into the fall, as I said, the two really outstanding resources, outstanding in their importance, dubious in their overall ecological value, are Clematis ternifolia, which is a relatively recently common and invasive anyway, though it's been here a while. This year I've seen it forming mats and tree canopies. It forms, it's on virtually every chain link fence in the city, I think. And it's still blooming now. It's too cold for anything to fly, but it's still blooming. And this is the flower of English ivy, heterohelix, or possibly hibernica. I can't really tell the difference. With, you'll note, a halictid native sweat bee on there, so it's not just honeybees we're talking about that are attracted to this. But alongside these, we did have a significant role, too, of our native Symphyotrichum, American asters, which are diverse and abundant. And as attractive as they were moderately attractive to honeybees in my study. I see them most frequently visited by bumblebees, and I think they're especially important for bumblebees in the fall. Finally, the pretty pictures that I promised, and Jen, I'm sorry these are not the ones from the Academy, but such pictures could be made with the Academy at the center. And instead, this is Richmond Library in Port Richmond. And this is the dance clouds that I talked about. So these are probability clouds. The hotter the color, the higher the probability that a spot was visited. And this is based on a small amount of data that a collaborator of mine, Carmi Oxman, who's a student at Drexel now, produced. I don't want to overinterpret this given the small size of the data set, but I do think it's significant that one hotspot emerges here that really stands out from the others. Does anyone know Port Richmond well enough to guess what that spot is? Got the Delaware River here. The bank kind of goes like this through that cloud. There's a very special feature of the landscape right there. My understanding is that it's been acquired by the city and will be a public park in the future. I hope that it is. This is Graffiti Pier. So this is I have not yet worked up the nerve to trespass on Graffiti Pier. There's a lot of really threatening signs and occasionally a cop car parked out front of it. Clearly, though, I would not be the first to do so. And I would do so with the best of intentions to survey the flora. I have suggested to the Botanical Club that as soon as we can go there without fear of legal repercussions, that we need to go and do a formal floral survey of Graffiti Pier. But this is what's attracting the attention of bees at Richmond Library. And this is a site that's essentially an abandoned post-industrial riverfront area that has been left to succession and become an early successional forest. And I think one thing this tells us, it suggests to us, is that there's a special ecological role for ruderal land in cities. Ruderal is a term that's used in landscape ecology. I believe it's derived from the word for ruin, but it describes a landscape that's derelict, that was something, but was kind of left to ecological succession. It's the wildlands of the city, which tend to be associated with economic decline. And certainly economic considerations need to be maintained. But I think that when urban landscapes start to become wild again, they perhaps take on a greater ecological function in some ways. And I think that without saying that we should let the land go, I think maintaining a certain wildness in designed landscapes is appropriate. And I hope that when this becomes a city park, that they don't turn it into a golf course with graffiti, but that it remains a forest and a parkland of some kind. More to come on that, I hope. Next steps. For some of you in the room, you might be really wishing I had not used honeybees in this study. I think there are good reasons for using honeybees, but there are also good reasons for not restricting our methods to honeybees. Bumblebees are just as amenable to pollen sampling and to colony weight monitoring as honeybees are. They don't dance, we can't do that, but as far as the weight data and the pollen data go, there's no reason in principle why a similar approach cannot use bumblebee colonies instead of honeybee colonies. This is a picture of Bombus fervidus, the golden northern bumblebee that I took this summer. I think it's my favorite bumblebee in this area. It's a really gorgeous bee. And it's covered with pollen that is so big you can see the individual grains with the naked eye, and that tells you with almost complete certainty that this is cucurbit pollen. It's been visiting a squash plant in my backyard, as it happens. This would also give us a chance to study floral resource niche partitioning. I mentioned earlier that honeybees are often the butt of jokes at best and the recipients of outright hostility at worst in ecological circles these days for their, especially in North America, for their potential to compete with or transmit diseases to native bees. While pushing back on that to some extent, I acknowledge that there's some legitimate concern there, and studying niche overlap between pollinators is central to the question of pollinator conservation vis-a-vis floral resources, and of disease transmission, because this occurs via floral resource vectors when pathogens are deposited on flowers. So it is relevant to be able to look at niche partitioning across pollinator species. In this case, this is a study by Leonard and Bluthkin from 2012 looking at honeybees and 2 species of bumblebee. And what you see is that there's overlap, but there's also noteworthy differentiation across these 3 species. But note the relatively low resolution we have here for pollen analysis, though. We have yellow pollen, dark brown pollen. And I say this not as a criticism. I've done microscopic pollen analysis, and it's hard, right? I've been in this boat too. I think we can, with molecular tools, we can achieve better taxonomic resolution and make more informative networks. And then finally, I've been working with Jason and some other folks in the entomology collection to try to do a pilot study of barcoding museum pollen residues that would enable us to go back in time and build historic plant pollinator networks. I'm not the first person to try this. This paper came out a couple years ago now, I think, just a year and a half ago. If you read the paper, the results were not a total success, but they definitely demonstrated that it's possible to amplify DNA from museum pollen. And so I want to go back and use what I think are a more refined version of the protocol and see if we can develop a really robust pipeline for analyzing museum pollen that would enable us to reconstruct historic plant-pollinator interactions. And thanks to Jason and the other folks in entomology, I picked pollen off of a bumblebee from 1870, I think, was the oldest one that I found. So if we can get DNA amplified off of that, then I think we can call that a success. I've not tried this yet. I have the samples, and I'm going to be heading to Penn State at some point this winter to run the lab protocol and see how it works. I promised to mention of iNaturalist too. This is the one of the projects that I administer, and like I said, there's nothing too special about being an administrator. You too can be an administrator on iNaturalist if you start a project. This is my most ambitious project, though, because it demands rather more of participants than most projects. And the goal of this project, it's inspired by Charles Robertson's monumental work in the late 19th, early 20th centuries, documenting plant-pollinator interactions in Carlinville, Illinois, by horse and buggy. And I think it's safe to say the vast majority of humans lack the genius that was Charles Robertson's, but we have something he didn't have, which is cameras in our pockets at all times and internet. And if we work together, I think we can do for whatever places we live in what Robertson did for Carlinville, Illinois, by documenting interactions between flower visiting insects and flowers. And so that's what this project does. What users do is upload. So the way iNaturalist works in general is you take a picture, ideally on your phone or something, with the geotagging enabled, so the geographic location is embedded in the photo file, and then you upload that, and then the public can contribute to the identification of it. In this case, I have people upload the observation, duplicate it, and then there's fields that allow you to link observations. So you link the flower to the bug to the flower, and then they are subject to community identification independently. So we have two observations, so that both the flower and the insect get equal taxonomic priority, and they are linked. And so what you get eventually is a data set of linked species interactions, which is a plant-pollinator network. And so theoretically, as observations accrue, no one is really competing with me yet, except for my buddy DJ here, who's just a little behind me in species. As observations accrue, we can, for given areas where we have a lot of observations, create plant pollinator networks from a citizen science data set like this. I want to acknowledge these folks. As I said, Carmi is a student here now, so I encourage you to introduce yourself if you get a chance to meet her. Both Ken Frank and Kirk Waddles are Philadelphia locals who have been really helpful in familiarizing me with local flora and just learning to think ecologically about the city. Tanya is my host here at the Academy. And then some of my Penn State colleagues, Christina is my postdoc mentor, and the man with the bees in the face, Don Shump. All right, thanks. Happy to take any questions.
In the image that you showed with the hot spot at graffiti here, there were 3 lesser hot spots.
Yeah.
Have you associated them with specific spots?
I only as much as I could do so with Google imagery, but they are green spots, and that's saying something in Port Richmond. So there's not a lot of green to be found here, and one interpretation of this is that the bees went out and found what green they could. So the other green habitats are basically open habitats. So what they're probably finding is clover that time of year. I think I saw your hand.
It looks kind of like an atom, distribution of electrons, but is that an artifact that there are 4 bulbs, or?
No, it's not an artifact. These are I forget how many dances went into this. This is a number of dances that have been decoded and plotted on top of each other. And so I think they were really going basically 4 different directions primarily. The shape that you see, so one modification I made to Roger's original script is I fit these with a kernel density function to give it a nice smooth look. It introduces an artifact in the sense that it's not intrinsic to the data, but I think it is intrinsic to the biology of the system that you have essentially a smooth probability function associated with the uncertainty of any given dance. So I saw your hand.
Yeah, there is sort of an idea, true or not, that vines are more present where there's less fire, warmer, fragmentation. And so do you think the current frequency of vine visiting reflects a historic change in abundance?
Yeah, I think that's the most, the finding that I find the most, that I spend the most time thinking about is the prevalence of vines that I saw, and the fact that prevalence seems to be relatively unprecedented in the literature. At least no one really talks about it. And I do think that it is when you do see it talked about, the context is almost always cities. Even though I'm not aware of anyone proposing vines as a characteristic urban flora, I think that's what we're seeing, that the urban disturbance regime, which is pretty much a fire-free regime. It's not a fire disturbance regime, but it is certainly still a frequent disturbance regime that keeps down canopy trees, and it really seems to favor vines. In addition, and not just exotic invasive vines like the clematis and the header we saw, but also the native vines like Parthenocystis, like Campsis, the trumpet vine. In learning the Philadelphia flora, as I said, mostly through riding my bike around the city, you just become really impressed by the ubiquity of these vines. Pretty much any surface that's not being mowed will have a vine on it eventually. Yeah, even some that are being mowed, vines will creep on. So I'll come back to you. Yeah.
So, well, looking at this diagram and thinking about maintaining this wildness in urban landscapes,
Yeah.
do you have an idea of how big a patch of wildness would be necessary? Like if we could convince everybody to leave 1 meter squared of your lawn, except in feet because we're in America, but like of your lawn wild, and you could mow the rest of your lawn, like is that helpful for bees?
So I think there are two questions embedded there. One is a question of how much is enough, and the other is a question of what spatial configuration is enough. And so, like, if everyone took a square meter of lawn and let it be wild, that would amount to a good chunk of land over the course across the whole city, but it would not be contiguous. So it would be just this highly distributed, highly fragmented configuration, which I think, I don't know what the ecological consequences would be, but surely it would be different than say, Lewisican Valley, which is this continuous tract of semi-natural land running through the city. My intuition as an ecologist is to say that anytime you can have contiguous natural land, that's better than fragmented natural land. For a highly mobile organism, like a flying insect, I think that the scale at which we consider fragmentation versus contiguity needs to correspond to the foraging scale of the organism. So for, say that little sweat bee that we saw here, these bees they're very small. They have a foraging range maybe a couple hundred meters. In that case, something the size of an athletic field is probably all the foraging habitat this bee is ever going to experience if it lives somewhere toward the middle of that habitat. Whereas honeybees, with the foraging size of square kilometers, we're talking like a municipal scale foraging environment, then their resource needs are much larger, but they're also they're able to find distributed resources very effectively. Most bees would fall somewhere in between. Well, most bees are small, but the bees that are not this small, the honeybee is the high extreme of foraging range. Bee this size would be on the lower end. A lot of bees fall in the middle. They can forage hundreds of meters, maybe up to a couple kilometers. I think, though, that flying insects really have an advantage over, let's say, ground beetles. Which some of them fly, but most of them they're fairly they're walking organisms, and for them, fragmentation, for organisms like that, fragmentation is a real problem. For flying insects, I suspect that they are more tolerant of it, which might be part of the reason why we see actually fairly diverse, in many cases, fairly diverse flower visiting insect fauna in cities. There's really interesting work coming out of the UK with Catherine Baldock and others that have found that in some cases you can even see cities acting as refugia of sorts for rare species that can't handle the different set of challenges that you have in like an agricultural landscape. But I think it's important not to, there's a tendency in some camps to view the city as this kind of paradise, pollinator paradise, where there's no pesticides and there's just lots of flowers. And in reality, there darn well are pesticides, and there may be lots of flowers, but most of them aren't attractive. So I think we need to think critically about urban landscapes while appreciating them where we can. Yeah.
One of the things that both agricultural landscapes and urbanized landscapes have done, and you alluded to this in the early part of your talk, is they've almost certainly altered the phenological discontinuity in floral resources for insects. And that affects different flower-associated groups of insects differently, depending on how specific and particular they are about the flowers that they visit. In the case of honeybees, they're probably much more adaptable than many of the other flower-visiting insects, and certainly more so than many flower-visiting Lepidoptera.
Yeah.
And one thing that has been noted by conservation biologists concerned about decline of rare or endangered butterfly species is that a key factor may be the appearance of periods of time in the normal adult foraging lifespan of an insect species where there aren't key flowers anymore that they depend on. Ironically, some of the situations where those gaps have been filled in result from the introduction of invasive flowers like non-native Cirsium species, for example.
Yeah,
And
or
you-
Aurelia. In
Or
our
Aurelia.
system.
But you have this kind of catch-22 situation where you have invasive plants that are having a negative impact, generally speaking, on the ecology of an area, but they may be essentially the lifeline for certain groups of plant-associated insects, where that phenology and continuous access to nectar sources has been disrupted by human activity.
Yeah.
How do you handle that in terms of planning restoration that's going to support maximum insect biodiversity in human-impacted environments?
This is arguably one of the most contentious issues in current conservation biology, and you can see on opposite ends of this polemic, we have folks like Doug Talamy on one end that are very focused on promoting natives over non-natives and present good evidence for doing that. And granted, he works mainly in suburban landscapes, kind of more moderately disturbed landscapes. And then you have folks like Peter Del Tredici from Harvard Forestry or something, who's a real advocate of weeds and the adventitious flora of disturbed landscapes, saying, look, these are the de facto native flora of these novel ecosystems that we've created where nothing else is really flourishing. And so if this is what will grow here, let's appreciate it for the function that it has. And I find myself convinced by both of them when I'm reading them, and obviously there's a dissonance there. I think there are no easy answers. I think you can, either extreme ultimately tends to reduce to absurdity. I think when there's this kind of laissez-faire, let evolution take its course, sure, it's invasive, but whatever, it'll be native in 1,000 years. That's a rather nihilistic view, I think, especially when you acknowledge the human responsibility for that introduction. I think there's a moral impetus on us to try to avoid the species extinctions that are bound to happen when we radically alter the biogeography of species. At the same time, though, something's value, ecological or otherwise, is not reducible to its geographic provenance, and I think you can just start listing counterexamples to that, which would include, for example, us. But also things like most of our major crops, most of our agricultural animals, and including the honeybee, though you can debate the ecological effects of agricultural animals. I think that you can veer off in this almost like it even takes on almost ethno-nationalist tone in some cases, so in this nativist position. I try to walk a careful middle line there and take species on a case-by-case basis. So, and also, you can describe and appreciate function without making normative statements about what should be done with a species when those statements can't really be made with available evidence. So, and it's okay to have a certain amount of contradiction. Japanese knotweed, one of the most destructive invasive plants in our area, is also one of the most important nectar sources in our area in the fall. I can appreciate the visitation by insects of Japanese knotweed. I can take pictures of it and say, wow, isn't this cool that this invasive plant has kind of filled the role that was left void when things like goldenrod were pushed out by the disturbance regimes that we have, while at the same time urging people to yank out knotweed whenever they see it. So, and a good example too is like, so Stiphnalobium and Lagerstromia, crepe myrtle, and Japanese pagoda tree, both of those are exotic ornamentals. Neither of them is widely regarded as invasive. And so I would not discourage people from planting these attractive both from a human and insect perspective trees as ornamentals, while at the same time I might tend to steer them towards something like Tilia americana as a native alternative. So don't plant knotweed. Maybe plant Japanese pagoda tree, but at a descriptive level, I think there's just a lot of empirical work to do.
Kind of two questions in one, based on literally what you just said. Do people, obviously people already give recommendations on what plants you should be planting for pollinators and like, you know, butterfly gardens and all this stuff, right? So that already kind of exists. Do you see like your research helping almost like define more what should be planted? Because like you were saying, like in the literature, it's like not really discussed as much. And then kind of that too, do you think your research could help like policymakers in the city government help when they're doing things like changing graffiti pier into a park, saying like, hey, these bee boxes on the library, you know, this is where they're going, and like if you change the park into golf courses, you know, grass, whatever, like that's a really terrible idea, and this is the reason why, and this is what you should be planting, yada yada yada.
Yeah, so the decision support angle that you're talking about was actually one of the explicit justifications for this project at the beginning. It was I wrote it into the grant as a justification for the work, and I really meant that when we understand the ecological function of plants in the city, we can make better informed decisions about how we manage our landscapes. And urban land is interesting in that it is highly discretionary compared to, say, agricultural land. If you're growing corn, then you need to grow corn. But if you have a postage stamp backyard in Philadelphia, you can do pretty much what you want with it as long as it doesn't annoy your neighbors too much. And so if we have a lot of discretion in how we use our urban green space, let's make as informed decisions as possible. And perhaps one of the most important choices is with the planting of street trees, which we do a lot of, right? Philadelphia is really rich in trees. There's a history of both good and bad choices made, though, with planting street trees. Like you see all these calorie pears that, first of all, they smell bad. They're exotic, invasive. They're covering roadsides and highway interchanges and stuff in a really a way that's ecologically damaging. But they're also visited by almost nothing. They're just not particularly attractive plants to foraging insects either. On the opposite extreme, we have American linden, which is also planted widely in the city, and it is maybe the most important nectar plant. During its bloom in late spring, it is probably the most important nectar plant blooming. And I'll also say from an apicultural perspective, it makes what I think is the most delicious honey that we have in the city. It's almost like a minty type of honey, surprisingly, from Tilia. So, yeah, the goal of this, what honestly I don't know as a scientist, is how to get the ear of the people making these decisions, and that's something I would welcome advice on. But I would love for decisions made by people managing land in the city, especially by the city itself that manages a lot of land, to be ecologically informed by this. And with Michael Nutter's Green Philly plan or something, the biological conservation is a stated management objective of Philadelphia land management now. So we need some criteria by which to make that decision, and the supporting of flower visiting insects is one legitimate and measurable criterion that we can work with.
Quick question, do you see Celastris? And a longer question, you have the seasonal pattern with peaks and gaps. One approach you could say is that's the way it should be and we should make sure, or you could say when we're planting we should really be finding things that bloom in early June and late June, early July to fill those gaps.
Yes, so very good questions. I admit I'm racking my brain. I know Celastris, but the common name is not coming to me. Bittersweet. Yes. Celastris shows up in my data, but in relatively trace amounts. And that can mean one of two things. It can mean it's not being foraged on, or it can mean it doesn't contribute much to pollen foraging, but it might still contribute to nectar foraging. The question, though, about, I'm glad you brought up the question of whether we should see this as like a natural pattern to be reinforced or a problem to be solved. So this pulsed pattern we see of resource availability. Something really interesting here is that this pattern of spring flows, dearth period, and I'm using apicultural lingo here, a flow, nectar flow when we're gaining weight, spring abundance, fall, late summer scarcity, and a brief fall flow. This is a pattern that is really archetypal across temperate systems from everything I can tell. There's, among beekeepers anyway, you hear this described in England, in France, in Germany, in Japan. It seems to be something that is driven by climate scale, like regional climate, more so than by the particular floral composition of a given place. And I think it's interesting that this archetypal pattern is preserved even in a landscape as radically altered as Philadelphia is. It seems that the regional climate and biogeography prevail over local effects in that regard. So then the question is, should this be reinforced or should it be fixed? One question I have is, so yes, this has been described all across the temperate world, but the temperate world is also the part of the world that's been most radically degraded. So it's basically urban and agricultural landscapes as this has been described, and I wonder to what extent this might be an artifact of human disturbance and not something that's really basic to like plant physiology or something. I suspect that while that might be the case to some extent, that there are probably good ecological evolutionary reasons for this pattern, though I'm not enough of a plant physiologist to suggest why, except that August is the hottest month of the year and flowers are expensive in terms of water. So maybe it's expensive to be a flowering plant in August. It is interesting, as Jason mentioned, that sometimes the things that fill these gaps are the exotic species that are basically divorced from any kind of ecological context, and they either just bloom whenever or they just don't stop blooming. And so you have something like white clover, which starts in May and blooms. It's still blooming. You can find it blooming outside now, though it finds its peak right in here. But then in this dearth period, the things that are filling this are the exotic crepe myrtle, exotic Japanese pagoda tree, exotic Aurelia. As we get into October, practically the only thing left in any abundance is exotic ivy. So, yeah, now this means different things for different species, though, and this is where I think we need to zero in on what our conservation priorities are. So for bees that have short flight seasons, or for insects in general with short flight seasons, which is the vast majority of them, what matters is what's blooming during their phenological active period. So for the spring andrena, for example, it really doesn't matter that there's an August dearth. However, for species that have full season active cycles like bumblebees and honeybees, those are the only bees that are quite that. I mean, honeybees are perennial, bumblebees are annual. In some of the social halictids, you still have fairly long active seasons, but they have to deal with that dearth somehow. And I think with honeybees, that's actually very much built into their biology. So when this dearth hits, this is about the time of year when bumblebee colonies are disintegrating and sending out reproductives, and that's about it for them. Like they've invested in their big pulse of reproductives, and then they're going to die anyway.
But their reproductive potential can be impacted significantly if that dearth is greater than
that is true. Yeah, if your new queens starve in August, then that's a real problem. So they need to make it at least until they go into diapause. So that is a good point. For honeybees, they can't dodge it in time, right? They're a perennial colony. They have to deal with it, and the way they deal with it, I think, pretty clearly from. You can ask any beekeeper what happens that time of year. They rob from each other, right? That's the time of year when honeybees start foraging on one another. Colonies will find weaker colonies and recruit to those instead of to floral resources. And that, I think, is one way to see that is like a natural population thinning. It's like a compensatory thinning of the population, because the colonies that are weak are not going to overwinter anyway. The colonies that are strong are going to get stronger by consuming the stores of their weak neighbors. And that's the fairly brutal economy of the honeybee colony in that time of year.
I think on that bombshell, we may have to wrap it up. Let's thank Doug one more time.